In this video, we'll have a look at what we call the endomembrane system. This is a number of organelles that are functionally or structurally related to each other. And in the endomembrane system, we're often looking at vesicles as being part of this system. So let's hop in and look at what are the components of the system. We have the plasma membrane, we have the nuclear envelope, we have the endoplasmic reticulum, Golgi, and the lysosome. So functionally they can be grouped together because of common function or because materials flow from one to the next to the next. Now let's start out, and remember, this is not a cell biology course, this is just a very quick snapshot. Let's look at the endoplasmic reticulum, and most of you have heard of this before, and you're aware that there's two types, the rough ER, and it's rough because ribosomes are attached onto the membrane, and we have smooth ER where there are no ribosomes, and we can see them represented here. So here is the rough ER. Once again, you can see ribosomes there. Here is smooth ER, and you can see that there are no ribosomes. You can also see that structurally they look a bit different. Here you have sort of flattened folds that lie on top of each other in the rough ER. Whereas when we get to the smooth ER, you can see it almost takes on a more tubular type shape. Now let's have a look at them. So the rough ER, right, what's it all about? It's most proteins enter in the rough ER or for transport out of the cell. So it's a lot about making stuff that I'm going to send out into the tissue matrix or to transport elsewhere in the body. The other thing is products enter the rough ER, and it also acts as a bit of a quality control that incorrectly folded polypeptide chains are detected and sent to the cytosol for recycling. So all of a sudden we have two things. It does quality control on proteins that are made, or polypeptides to be more correct, and also it's about transporting proteins out. Now, packages of these secretory proteins are put into transport vesicles, and these are made of a phospholipid bilayer, and they're sent to the Golgi apparatus for further processing. It also has another role, that it produces membrane components for many of the organelles that depend upon having a membrane, and also for the plasma membrane. And if you think back about our discussion about the plasma membrane, remember a lot of it was phospholipids, but there's a lot of proteins that are like transmembrane proteins, peripheral proteins, etc. So a lot of those integral or peripheral proteins are produced in the rough ER and eventually transported to be incorporated into the plasma membrane. Smooth ER, no role in protein synthesis, so it does something different. It can store calcium ions that are pumped out into the cytosol, and basically it's like a bank or a bank account where you can put calcium in there for later use. We'll see when we get to skeletal muscle cells that they use a modified form of smooth endoplasmic reticulum to store calcium ions that will aid in muscle contraction. Also, detoxification reactions. So it helps to limit the damage by certain chemicals that could be in the body or substances. And the next thing is lipid synthesis. So this is where most of our plasma membrane phospholipids and the cholesterol, also a number of lipoproteins and steroid hormones, are produced in the smooth endoplasmic reticulum. So you can see it actually has quite a number of different tasks. Here we can just see sort of the relationship. You can see there's the nuclear envelope being cut away. You can see the rough endoplasmic reticulum. You can see the various ribosomes on there. And you can see here smooth endoplasmic reticulum. Of course, there are no ribosomes on there. Now, the Golgi apparatus, when we see, let's back up. No, they don't show it. They show it right here. You can see there's the Golgi right there. So you can see between the ER and the membrane, the Golgi apparatus is usually sort of just stuck in there. And the Golgi apparatus we find between the rough ER and the plasma membrane. And have a look at it. To me, it kind of looks like a bunch of pancakes that are kind of stacked up on top of each other. So they're flattened membranous sacs filled with enzymes and other molecules. So what do we have happening here? Well, proteins and lipids made by the ER are modified. They're sorted and they're packaged for export in the Golgi. So the Golgi isn't necessarily making them, but it's really about modification, package, and getting it ready for shipping. Now, where are these products going to go? So the products packaged by the Golgi can be secreted from the cell through exocytosis. They may end up as part of the plasma membrane, or they could get sent to the lysosome. Okay. And here we can just see a picture of the Golgi, and you can see, first of all, its location. As I said, it's usually sandwiched between the endoplasmic reticulum and the plasma membrane. You can see that, yes, vesicles from the rough endoplasmic reticulum will travel and become incorporated into the Golgi, where the material will be modified and packaged. And then the finished product is packaged at the other end of the Golgi for the various sites where it may head off to, which we just talked about. Have a look. You can read about cystic fibrosis. I think I'm not going to spend much time on here, not because it's not important, but I think you can have a look at it. You'll also find this material in the book, and it's a good way to sort of link your knowledge of the organelle with the pathophysiology of a disease on a molecular level. Okay, lysosomes. And these organelles are interesting because they're basically about digestion of worn out cells or cellular components. So they do a lot of sort of cleanup, a lot of getting rid of the waste things that have been basically used up. And so, not surprisingly, it contains digestive enzymes like acid hydrolases. And what happens is it takes a lot of these worn out or no longer needed macromolecules and breaks them into subunits. And what happens? Well, they could get released into the cytosol for dispersal, or it may get recycled into new macromolecules. And you can see lysosomes are fairly small in size compared to things like the mitochondria or the Golgi that we can just see sort of right there in the bottom. So, just sort of remind ourselves that this whole endomembrane system, you can walk your way through here. Like smooth ER makes lipids, rough ER makes proteins, and each product is packaged in vesicles to be transported to the Golgi. What does the Golgi do? Sorts it out, further modifies both lipids and proteins. It packages, and there's three possible pathways for things once it leaves. It could be sent to a lysosome to be involved in basically those catalytic reactions. It could end up in the plasma membrane or in the membrane of other organelles, or it could be sent to the plasma membrane where through exocytosis it is secreted. So that's a quick look at the endomembrane system. And once again, the book is great. Read the book. That's where you'll get another telling of the story, and it really helps to reinforce your knowledge. And also the book's great because not only does it talk about each thing individually, it comes back and will give you these summaries, these really nice summaries. And once again, remember, it's all about coming back to information to make sure that with repetition it becomes part of your knowledge bank. Okay, we're going to end the video there. Quickly look in your book at some of the lysosomal storage diseases, such as Tay-Sachs, which I think we have talked about in class, Hurler's, and Niemann-Pick disease. Just give you an idea of how these organelle or lysosomal storage diseases function within the body.